Good day, everyone. I am Angelica from the Bachelor of Secondary Education, major in Sciences. And today, I will going to discuss my topic which is all about the membrane proteins. So, here's our introduction. Biomembranes are crucial components in the genesis of life and the structuring elements of the organization of living cells. They preserve the functional integrity of the cell organelles and regulate the exchange of solutes and signals between the different functional areas of the cell. So membrane proteins, which constitute about 30% of the entire protein content of the cell, are often very complex and function in many different ways. A large number of today's drugs are targeted as membrane proteins. In spite of their importance in modern life sciences, the structure and the function of the membrane proteins are not well understood, and only few membranes proteins have been investigated in detail. So lipids in the membrane make a hydrophobic barrier for aqueous solutes. So this barrier is semi-permeable due to the presence of membrane proteins. In addition to transport functions, membrane proteins have roles as receptors, enzymes, and structural components, and the myomembranes are also contain glycolipids and glycoproteins whose carbohydrate constituents are external of the cell and are important in recognition, according to Klein Meet in 2003. So membrane proteins are essential to all forms of life. Millions of membrane proteins are found in the lipid membrane layer that surrounds cells and in the lipid membrane layers that surround smaller cellular compartments. So many medicines interact with the membrane proteins. So these include drugs that treat cancer, heart disease, and pain. Research into membrane proteins is therefore important to the design and development of new medicines. So membrane proteins are difficult to work with, partly because they are also small. However, using techniques such as X-ray, crystallography, and electron microscopy rather, structural biologists, including those at the membrane protein laboratory, are able to see the atomic detail of membrane proteins. So, there has been great progress in the field of the membrane protein and its structural biology over the past 15 years. So, in 1988, Harmu Michel and Johann Disenhofer and Robert Huber received a Nobel Prize for the first crystal structures of membrane proteins. So those from the photosynthetic reaction center of Rhodosodomonas viridis, in which Rhodospiodomas viridis has been renamed Blastochloris viridis. So, over the next 2 decades, new membrane proteins crystal structures were sufficiently rare to the grace of covers of nature and science. When they did appear, and however, their number rose exponentially, so the database now contains more than 650 unique structures of membrane proteins determined with X-ray crystallography or nuclear magnetic resonance or the NMR spectroscopy, and three of the good overall agreement in structures that have been solved by both. So, the availability of the detailed membrane proteins structures allows researchers to further probe their function and dynamics using genetic, biochemical, biophysical, and computational tools. Although the basic structure of biological membranes is provided by the lipid bilayer, membrane proteins perform most of the specific functions of membranes. So it is the proteins. It is the proteins. So therefore, it gives each type of membrane in the cell and its characteristic functional properties. So accordingly, the amounts and the types of proteins in the membrane are highly variable. So in the myelin membrane, which serves mainly as electrical insulation for nerve cell axons, is less than 25% of the membrane mass is protein. So, by contrast, in the membranes involved in the ATP production, such as the internal membranes of mitochondria and chloroplast. So, approximately 75% is protein. An atypical plasma membrane is somewhere in between, with the protein accounting for about 50% of its mass. So what are membrane proteins? So biological membranes consist of double sheet known as a bilayer of lipid molecules. So this structure is generally referred to as the phospholipid bilayer. In addition to the various types of lipids that occur in the biological membranes, membrane proteins and sugars is also the key components of this structure. So when we say membrane proteins, these are play a vital role in the biological membranes as they help to maintain the structural integrity, organization, and flow of material through membranes. And sugars are found on one side and bilayer only, and are attached by covalent bonds to some lipids and proteins. So there are three types of lipids. These are the three types of lipids that can be found rather in the biological membranes, namely phospholipids, glycolipids, and sterols. So when we say phospholipids, they are containing glycerol that are referred to as glycerophospholipids. It as an example of glycerophospholipid that is commonly found in biological membranes is phosphatidylcholine. So which has a choline or choline molecule attached to the phosphate group. So when we say also serine and ethanolamine, it can also replace the choline in this position. So and these lipids are called phosphatidylserine or PS or the and the phosphatidylethanolamine phosphatidylethanolamine rather or the PE respectively. So phospholipids can also be sphingospholipids that is based on the sphingosine and such as the sphingomyelin. So the next one are the glycolipids and the third one is the sterols. So when we say glycolipids, it can contain either glycerol or sphingosine and it always have a sugar such as a glucose in place of the phosphate head are found in phospholipids. So when we say also sterols, they are absent from most bacterial membranes, so but are an important component of animal or typically in the cholesterol and mainly in plant it is found in the stigmasterol membrane. So cholesterol has a quite different structure to that phospholipids and glycolipids. So it consists of a hydroxyl group which is hydrophilic head region, so a four-ring steroid structure and a short hydrocarbon side chain. So here are the first subtopic of the membrane proteins. The amphipathic lipids formed by layers. So all membrane lipids are amphipathic, that is, they contain both a hydrophilic or which is what we call the water-loving region, and the hydrophobic, which is the water-hating region. So thus, the most favorable environment for the hydrophilic head is an aqueous one. So whereas the hydrophobic tail is more stable in the lipid environment. So the amphipathic nature of the membrane lipids means that they naturally form bilayers in which hydrophilic head point outward towards the aqueous environment, and the hydrophobic tails point inward towards each other. So when placed in water, membrane lipids will spontaneously form liposomes, which are spheres form a bilayer with water inside and outside by resembling a tiny cell. And this is the most favorable configuration for these lipids, and as it means that all hydrophilic heads are in contact with water and all of the hydrophobic tails are in lipid environment. So, in early experiments by Gorter and Grendel in 1925 were the first to demonstrate the biological membranes are bilayers. So these researchers extracted the lipids from red blood cells and found that they occupied a space that was twice the surface area of the cells. So red blood cells contain no internal membrane, so they deduced the plasma membrane must be composed of two layers of lipids. So the next subtopic, which is all about the biological membranes and the fluid mosaic. So the fluid mosaic model proposed by Jonathan Singer and Gart Nicholson in 1972 that describes the dynamic and fluid nature of the biological membranes. So the lipids and proteins that can diffuse laterally through the membrane. So phospholipids can diffuse relatively quickly in a leaflet of a bilayer in which they are located. So a phospholipid can travel around the perimeter of our red blood cell in around 12 seconds or move the length of the bacterial cell within 1 second. So phospholipids can also spin around on their head to tail axis, and their lipid tails are very flexible. So these different types of the movement create a dynamic. So fluid membrane, which surrounds cell and organelles, and membrane proteins can also move laterally in the bilayer, but their rates of their movement vary and are generally slower than those lipids. In some cases, membrane proteins are held in the particular areas of the membrane in order to popularize the cell and enable the different ends of the cell, which has two different functions. And one of the example of this is the attachment of glycosyl phosphatidylinositol, or the GPI, which anchor to proteins to target them to the apical membrane of epithelial cells and exclude them from the basolateral membrane. So as you can see, fluorescence photo-bleaching is one of the experimental methods that is used by scientists to demonstrate visually the motility of proteins and lipids in a bilayer. So a lipid or membrane protein located in the surface of the cell, which is tagged a fluorescent marker such as green fluorescent protein or GFP. So a beam of a laser is then focused to a small area of the cell surface using a fluorescence microscope in order to bleach the fluorescent tags in this area so that they no longer emit a fluorescent signal. So this small area of membrane is absorbed over time, and gradually the fluorescence increases again, so indicating that other tag proteins or lipids are diffusing into this region from elsewhere in the membrane. So this demonstrates that the lipid bilayer surrounding cells is fluid in nature and allow lateral diffusion of the both lipid and the membrane proteins. So, how membranes are made? So biological membranes are formed by adding to a pre-existing membrane. So in eukaryotes or prokaryotes rather, this occurs on the inner leaflet of the plasma membrane facing the cytoplasm, and in eukaryotes, membrane synthesis takes a place at the ER or the endoplasmic reticulum on the cytoplasmic leaflet of the endoplasmic reticulum membrane, as termed as the inside of the cell. So lipids then leave the endoplasmic reticulum and travel through the secretory pathway for the distribution of the various subcellular compartments of the plasma membrane. So in eukaryotic cells, enzymes that span the endoplasmic reticulum catalyze the formation of membrane lipids. So in the cytoplasmic leaflet of the endoplasmic reticulum membrane, two fatty acids are bound, so one by one, to glycerol phosphate from the cytoplasm. So this newly formed diacylglycerol phosphate is anchored in the endoplasmic reticulum membrane by its fatty and acid chains rather. So the phosphate is then replaced by the head group, which is such as the phosphate and the choline. So flipases in the endoplasmic reticulum membrane can then move some of these newly formed lipids to the luminal side of the endoplasmic reticulum membrane. So similarly, flipases in prokaryotes can transfer new lipids from the inner leaflet of the plasma membrane to the outer leaflet. So these flipases are responsible for adjusting the lipid composition of each layer of the membrane. So in eukaryotes, lipids must then be distributed to the various intracellular membranes. So the traffic of the vesicles between organelles and in combination with signals that direct particular lipids to the specific locations is required to create a correct lipid composition in all of the cellular membranes. So vesicles bud from the endoplasmic reticulum and travel via endoplasmic reticulum, Golgi intermediate compartment, or the what we call the ergic, to join the Golgi, where sorting of lipids takes place. So as you can see, the Golgi then sends lipids in vesicles to the various destinations, including the plasma membrane and lysosomes, and lipids and proteins are internalized from the plasma membrane into endosomes. So organelles such as mitochondria acquire the lipids from the endoplasmic reticulum by a different mechanism. So water-soluble proteins called phospholipid exchange proteins remove phospholipids from the endoplasmic reticulum membrane and deposit them in the membranes of the appropriate organelles. So the next subtopic, which is all about the synthesis of the membrane proteins. So in eukaryotic cells, the synthesis of the membrane proteins destined for the plasma membrane, endoplasmic reticulum, or any other membrane-bound compartment begins to cytosolic ribosome. So after a short segment of the protein has been synthesized, the ribosome and the messenger RNA and nascent protein chain associate with the endoplasmic reticulum, where the rest of the protein is made and simultaneously inserted into the membrane. And this phenomenon was first explained by Gunther Blobel, David Sabatini, and Bernhard Doberstein in the 1970s, and these scientists proposed that there is a binding factor which recognizes the emerging protein chain and can dock the ribosomes at the endoplasmic reticulum membrane. So this is what we call the cold translational targeting. We have this cold translational targeting. It is a dominant mechanism for protein delivery to the endoplasmic reticulum in higher eukaryotes, whereas yeast and prokaryotes favor post-translational targeting, so whereby proteins are delivered to the endoplasmic reticulum after completion of synthesis. Second, the post-translational targeting. So it also occurs in the higher eukaryotes. So often when a membrane protein is so small that the signal sequence that does not emerge until the whole protein has been synthesized. So post-translational targeting can be carried out both of SRP dependent by SRP independent mechanisms. So here are the classes of membrane proteins. So membrane spanning proteins are diverse in structure and function. So they can be constructed as the A helices or from the B barrel. So the B barrel membrane proteins often function as pores and hydrophobic amino acids facing out into the bilayer, and in addition, there are other non-spanning proteins which associate with a bilayer, often using the hydrophobic anchor. And on the other hand, this is also what we call a helical membrane proteins. These are the proteins that have been listed one a helical hydrophobic stretch of amino acids around the 20 residues in length, which corresponds to 30, in which 30A or the thickness of an average phospholipid bilayer. So if an ahelical membrane protein spans the membrane for more than once, it will have more than one these hydrophobic sections. So for example, the Ka2 plus ATP ATPase of the endoplasmic reticulum and the sarcoplasmic reticulum spans the membrane 10 times, so it has 10 hydrophobic stretches that around the 20 amino acids each. So this is also, we have also what we call the passive and active transport in membrane proteins. So when we say passive transport, it can also be mediated by carrier proteins that carry specific molecules such as amino acids down concentration gradient, again without any requirement for energy. So we have also here the active transport. It moves species against concentration gradients and requires the energy which is obtained from ATP from light or from the downhill movement of the second type of molecule or ion within the same transporter. So here are the functions of the membrane proteins. So the tremendous progress in the structural biology of the membrane protein, supported by the countless genetic, biochemical, and biophysical studies that are now provides exciting details of the different molecular functions. So specificity of channels is illuminated by their architecture and gating mechanisms. So fundamental transport mechanisms are shared among transporters of widely varying families, so capturing intermediates in the reaction cycles of membrane enzymes provides the details of their mechanism. So by capturing the intermediates in the reaction cycles of the membrane enzymes, that provides details of their mechanism. So an atomic picture of a receptor interaction with signaling transducers provides vital clues to the molecular transmission of the information. So before looking at the examples of these membrane functions, it's also worth remembering the classes overlap. So for example, many receptors from ion channels and some enzymes also transport substrates or ions. So here are the last topic, which is all about the membrane protein complexes. So a membrane protein complex composed of 2,000 amino acids in 14 polypeptide chains in shown in the high resolution structure of complex 1 from T, so or the thermophilus. So the overall shape and subunit interaction suggests a coupling mechanism in which the lateral helix acts as a piston to drive the conformational changes that open proton channels in response to the reduction of the bound Kinon or Q. So according to Sanders and Meir in 2004, it stated that medical genetics so far has identified 16,000 missense mutations leading to single amino acid changes in proteins sequences that are linked to the human disease. So a majority of these mutations affect folding or trafficking rather than specifically affecting protein function. So many disease-linked mutations occur in the integral membrane proteins or in which a class of proteins about whose folding we know for a very little. So here are the published studies about the membrane protein. So according to MacGregor, Chen, Pomeroy, Wang, Go, Chakrabarty, and Pry in 2003, they stated that structural study of the membrane proteins requires detergents that can effectively mimic lipid bilayers, and the choice of their detergent is often a compromise between detergents that promote protein stability and detergent that form small micelles. So, researchers describe lipopeptide detergents or the LPDs, in which it is a new class of amphiphile in which it consists of a peptide scaffold that supports the two alkyl chains and one anchored to each end of an A helix. So the goal was to design a molecule that could self-assemble into a cylindrical micelle with a rigid outer hydrophilic shell surrounding an inner lipidic core. So consistent with this design, LPDs or the lipopeptide detergents self-assemble into a small micelles that can disperse phospholipid membranes and are gentle, non-denaturing detergent that preserve the structure of the membrane proteins in the solution of the extended periods of time. So the LPD design allows the membrane-like packing of the alkyl chain in the core of the molecular assemblies and possibly explaining their superior properties relatively to the traditional detergent in stabilizing membrane protein structure. So the other one we have here, according to Stroop and Brunger, in 2005, they described that the concentration of the detergent in membrane proteins preparations can have a critical role in protein stability, function, and the potential for the crystallization. So this method was applied to quantitate the amount of the detergent remaining into a solution after concentration in various concentrators. So researchers found that ability of the tested detergents to pass through the molecular weight cutoff membrane correlates well with detergent micelle size. So therefore, the micelle size can be used as a rough guide to estimate the retention of a given detergent in various molecular weight cutoff concentrators. So the refractive index method is exceptionally informative when coupled with the size inclusions of chromatography and light scattering and can be used to determine the oligomeric state of the membrane protein in which the size of protein associated micelle as well as the amount and the size of unbound detergent micelle. So according also to Herkman and Tanaka in 1986, they also described that a plasma membrane enriched fraction prepared from barley roots was analyzed by two-dimensional gel electrophoresis. So four methods of sample solubilization were assessed on silver-stained gels. So when membranes were solubilized with 2% sodium dodecyl sulfate followed by addition of nonylate P40, gels had high background staining a few proteins because of incomplete solubilization. So gel of membrane solubilized in urea in nonylate P40 that had greater number of proteins, but proteins with molecular weight with greater than 85,000 were absent, and proteins with low molecular weight were diffuse. So high molecular weight proteins were present in gels of the membrane solubilized in 4% and sodium dodecyl sulfate followed by acetone precipitation, but background staining and streaking remained a protein. So gels of the best quality were obtained when the membrane proteins were extracted with the phenol and precipitated with ammonium acetate in methanol. So background staining and streaking were diminished and proteins were clearly resolved. So these are these topics. This is the special topic which is all about the viruses with emphasis on the SARS-CoV-2 and causes of the disease COVID-19. So the current coronavirus disease in 2019, or what we call the COVID-19 pandemic, has triggered unprecedented collective research efforts from the scientific community to better understand the disease and its cellular and molecular mechanisms to identify efficient therapeutic drugs for taking care of infected patients with the severe acute respiratory syndrome coronavirus or the SARS-CoV-2, and to develop the vaccines for protecting the whole population from the infection. It is cited according to WHO, Poland, and Zachary in 2020. So one of the initial challenges in the fight against this virus was to rapidly detect SARS-CoV-2 infected patients to limit propagation of the virus through isolation, according to Ravi in 2020, and another challenge was to better understand the global antibody responses against SARS-CoV-2 proteins in patients, according to Poland, Zohar, and Alter in 2020. So, among the antiviral immune responses, it elicited in infected patients or immunoglobulin or the IG. It responses against the viral transmembrane proteins in which it expresses the surface of the virus envelope that are important for generating antibodies that limit virus propagation. So this occurs by preventing interactions with host cells, such as production of neutralizing antibodies that block the binding of the viral transmembrane spike protein to its receptor or angiotensin converting enzyme 2 or the ECE2. So it expressed by infected host cells, according to the Poland, Land, Hoffman, Zohar, and Alter in 2020. So, some of these cellular actors, such as macrophages and neutrophils, could also contribute to the aggravation of the disease by releasing chemokines or cytokines that enhance inflammatory cascades described the cytokine storm. So, leading the lesions of the infected tissues, although the involvement of antibody is dependent mechanisms still need to be confirmed in patients with COVID-19, according to Zohar and Alter in 2020. So most of the serological assays developed against the SARS-CoV-2 are based on the recognition of the viral transmembrane spike protein and the nucleocapsid protein N. So it considered as major targets of the responses. So besides from the viral spike protein, little is known on the Ig responses toward the others viral transmembrane proteins E and M also directly exposed to the host immune system. So spike, the envelope, and membrane are integral membrane proteins that transit through the host cells to the whole cells of the endoplasmic reticulum and the intermediate compartment or the ergic, and most likely to the Golgi complex in the endoplasmic reticulum, and these proteins are N-glycosylated, folded, and assembled in ergic for virus budding and release, according to Sikari in 2020. So this maturation process is the key for proper viral protein function. So for instance, spike and link glycosylation is required for the virus entry into the host cells, impacting directly on spike stability during its synthesis. So instead of its binding ability of the ACE2 receptor, according to Young in 2020, and these modifications might be also key for antibody recognition. So it is in the present study, researchers relied on an experimental systems that recapitulates protein modifications and acquired through the host cells secretory pathway to explore the antibody responses of SARS-CoV-2 infected patients. So researchers found that S and M proteins, but not E, are exhibited antigenic domains recognized by IgG, IgM, and IgA in SARS-CoV-2 infected patients. So these are also high levels of IgA or responses were observed in the patients with COVID-19 with the moderate and severe forms of the disease. So finally, SARS-CoV-2 spike D614 and G614 variants were compared, so showing those of Ig binding on the spike of G614 variant. So altogether, this study underlines the necessity of considering the mammalian cellular system to better characterize the serological status of the patients with COVID-19. So how COVID-19 related to the educational sector of the system of the country. So in December 19, an outbreak of novel coronavirus is known as the COVID-19 occurred in the China that has spread rapidly across the globe within the few months. So COVID-19 is an infectious disease caused by the new strain of coronavirus that attacks respiratory system, according to the World Health Organization in 2020. So as of the January 2021, COVID-19 has infected 24 million people and has caused 2 million deaths in 191 countries and territories, according to Johns Hopkins University in 2021. So this pandemic has created a massive disruption of the educational system, affecting over 1.5 billion students. So it has forced the government to cancel national examinations and the schools to temporarily close, cease face-to-face instructions, and they strictly observe physical distancing. So these events have sparked the digital transformations of the higher education and challenged its ability to respond promptly and effectively. So schools adapted relevant technologies and prepared learning and staff resources. So it set systems and infrastructure, and this can also establish new teaching protocols and adjusted their curricula. However, the transition was smooth for some schools but rough for others, particularly for those from developing countries and limited infrastructure, according to Pham and Nguyen Simbulan in 2020. So, the COVID-19 pandemic has affected educational systems worldwide, leading to the near total closures of schools, universities, and colleges. So most governments around the world have temporarily closed due to the educational institutions to contain the spread of the virus. So these nationwide closures are impacting over 60% of the world students' population, and several other countries have implemented localized closures affecting the millions of additional learners. So studies on the impact of COVID-19 pandemic were conducted in the Philippines, which not just clearly revealed strong reactions among students but also discovered teachers' level of anxiety and amid school closure. So however, Filipino teachers' positive outlook amid psychological stress or anxiety was seen. So teachers indeed had to face work and life changes during the pandemic situation. So and moreover, teachers faced challenges with the online blended learning approach as they embrace the new normal in teaching. So the are still challenged to fill the gaps in providing information, communication, and technology or the ICT resources and the capacities of both teachers and students as they embrace the paradigm shift in pedagogical delivery. So relatively, the work commitment of the organization's employees is amidst the crisis like a pandemic that may be affected. So when an emergency occurs with an organization, it usually disrupts daily activities. So it can generate fear of the financial loss of its impact to the organization that could be immediate or long term. So as Philippines combat against the COVID-19 pandemic, the Department of Education started to brace the new normal education like what I've said before amidst the pandemic situation in the country. So DepEd conducted a nationwide consultation with stakeholders and education experts and prepared for learning continuity plan to deliver education to learners. So in preparation of school year 2020 and 2021, DepEd are was provide a self-learning modules or SLMS. So with the alternative learning delivery modalities for various types of learners across the Philippines, including modular television-based, radio-based instruction, blended and online. So SLM are delivered in printed format to schools located in coastal areas or far-flung provinces and communities without access to the internet or electricity. For households with gadgets and devices, the department has announced the SLM can also be accessed online or offline. So inevitably, schools and other learning spaces were also forced to migrate to the full online learning as the world continues to battle the control of vicious spread of the virus. So online learning refers to the learning environment that uses the internet and other technological devices and tools that synchronous and asynchronous instructional delivery and management of academic programs according to Asher and Barak in 2020 and Wang in 2019. So synchronous online learning involves a real-time interaction between the teacher and the student. So while asynchronous online learning occurs without strict schedule for different students according to Singh and Thurman in 2019. So, how COVID-19 related to the educational sector? So, with the reference to policies, government education agencies and schools scrambled to create foolproof policies in governance structure, teacher management, and student management rather. So, teachers who were used to conventional teaching delivery were also obliged to embrace technology despite their lack of technological literacy. So to address this kind of problem, online learning, webinars, and peer support systems were launched. So on the part of the students, dropout rates increased due to the economic, psychological, and academic reasons. So academically, although it is also virtually possible for students to learn anything online, learning may be perhaps be less than optimal, especially in courses that require face-to-face contact and direct interactions, according to franchise in 2020. So, I think that's all. Thank you for listening, and God bless.